Good morning, everybody, and thank you very much for joining our SPHN webinar today. We will hear about the SPHN Infrastructure Development Project today, SVIP, the Swiss Variant Interpretation Platform, a project funded jointly by SPHN and Biomed IT in 2017 and 18 it started. SVIP aims at establishing a one-stop shop for clinical interpretation of variants in oncology. And this curated database will enable clinicians to base their diagnosis on the same high quality information that is also been made available to researchers. We are now very curious to hear about the progress of this project. And with this, I would like to welcome our speakers of today, Valérie Barbié and Daniel Stekhoven. Valérie is the Director of Clinical Bioinformatics at SIB, the Swiss Institute of Bioinformatics in Geneva. She's a computer sciences engineer by training and has worked in the pharmaceutical industry for many years before joining SIB in 2015. Daniel Stekhoven. Daniel is the co-director of the NEXUS Personalized Health Technologies core facility at ETH in Zurich, where he is leading the clinical bioinformatics unit. He's a mathematician by training and founded a company for statistical consulting before he was asked to set up NEXUS in 2014. Since 2016, he's also a group leader at the SIB. Swiss Institute of Bioinformatics. So I'm very happy to pass the word to you, Valérie and Daniel. Thank you very much for taking the time and being with us today. The floor is yours.
Thank you very much, Catherine, and thank you for the opportunity to present our project. Once again, as I may say, we did a presentation before in the DCC webinar, as it was called back then. And I hope we can give you additional and interesting details on what we've been up to in the last couple of years. So I will, if you maybe not have attended the first webinar, which you should, but I will give you still a brief background on how we ended up doing what we're doing. So there are a lot of available places to get information about variants in cancer and how to interpret them and ultimately how to report them to clinical end users. The problem is finding the needle in the haystack, and there is this still very good figure from Good showing that with the dawn of new technology, namely next generation sequencing, the data simply got even bigger. And you can see this funnel here where the data is getting smaller and smaller and smaller. At one point, we are annotating and make functional predictions. But then somewhere here is this poor person who needs to put the last evidence pieces together and then write a report to enable clinical application. So we are bioinformatician, you heard, computer sciences engineer and a mathematician. What do we know about clinical interpretation in cancer? So we asked the experts what they need. So the SIB set out, they formed a working group for somatic mutation calling. We invited all the university hospitals and beyond. We had many meetings with them. This was when meetings still were a thing, you know. And we asked them what they need. And we came up with a wish list. The first thing they said is, we want to have a joint repository at one place where we can go to and look stuff up. And of course, everybody wants to help the greater good in enabling the battle against cancer, but clinicians don't have time to do that. So it would be great if you could post variants for everybody to see. But you know, if I look up a variant in some database, I would be so great if somebody could tell me, somebody I trust, somebody who is maybe another medical expert in Switzerland, whether this is a trustful annotation or not. So they want trusted validation. They want to actually approve that one certain variant in a certain gene for a certain disease means one thing and one thing only. So there needs to be approved, mutually approved annotation. If there is no agreement, then we need a way to find, to manage discrepancies between interpretations. And ultimately, variant annotation is a moving target. So interpretations and annotations do change over time. And wouldn't it be great that this one variant, where you didn't know what it meant for treatment, got suddenly presented with a new clinical trial showing actual efficacy of a new drug, and then you get notified about this? So notification was at the end of the wish list. So what does that mean? We took this wish list and we planned to build a system that allows in a front end, in a web-based front end manner, to submit variants, to curate variants. So basically add a first draft annotation based on public evidence, review these annotations by experts, and ultimately a retrieval interface where you can look up these variants. In the back end, as I said, public evidence need to be stored somewhere. We want our own annotations stored in one place. And what sets us a bit apart from other variant annotation databases, we need variant data essentially in SVIP, not only to have a proper curation process and to see additional information, like for instance, variant frequency, but also to enable the communication or the notification process, that we know from this hospital, we have had a variant submitted earlier, so they might be interested in being notified about it. And ultimately, the user management. Now you can imagine there is the output is reporting. So basically the pathologist or the hemato-oncologist who is doing the interpretational work, they ideally want an easy to use report coming out of the system. Notifications are managed and security. And there are some dependencies between these elements. So starting to build this system was quite a challenge. But let me hand over to Valérie now, and she's going to tell you something about, yes, security.
Thank you, Daniel. As Daniel just mentioned, actually, the IT infrastructure was only one of the numerous challenges we had to cope with, but it was definitely complicated, and obviously we didn't do this ourselves. We had a lot of experts around us, IT and IT security guys. And the reason why it was complicated to put down is that on one side, we have to deal with different profiles who need different things from the system. And on the other side, we deal with highly sensitive data that obviously we need to protect because these are patient-related information. So we'll start with one thing, the part that is called the public access side. So this part, next slide, please, Daniel, is really intended to serve research and even clinical research, but people who are not partners of SVIP. It means there is an open web page, no login required, nothing. It's completely fully available. And everybody is logging through accessing the system through the page, SVIP.ch. And we have set up something with, you see what we call a DNS load balancing. So basically we have two instances of this, one located at ETH Zurich and the other one located in Lausanne at SIB. They are mirrored. They are exactly the same system, exactly the same content. The objective was simply to ensure that we have a good system availability. If one is down, the other is taking over. We pretend that we want to serve the clinical research world, so we don't want people to be out of their system for 1 week, especially because you will see we provide an API so that additional external resources can query SVIP, and it's very bothering when the application does not respond. So this is the public side. It contains only variant information, and Daniel will demonstrate this. There's absolutely nothing sensitive and nothing patient related. On the other side, we have the biggest part, which is what we call our private infrastructure. So basically, it's intended to serve our curators who are internal to SIB, and our data providers, basically our clinicians who are the partners in the project. So you see they have the same entry point. It's a different address. It's a different system technically. Same entry point, but obviously different rights. Curators don't do the same actions as our clinicians, that's clear. The reason why our curators are on this side, it's because on the right side of the slide, you have people who need to write information into the system, while on the left side, it's only people who can see information. This is why we have the curators on the same side of the data providers. And basically what you see, first we have a zone that we call an access zone, which is somehow a buffer. This is the place where people can connect and where also the data can be transferred directly by the data providers. This transfer is automatic and encrypted, of course, for security reasons. And below this, next slide, we have an additional infrastructure, which is called the restricted zone, which is basically the one who is handling everything, the backend, the complete database, including sensitive and non-sensitive information. And you see the key concept here is that the data, everything that is sensitive is never in contact directly through the outside world. It's only through the access zone, which is adding an additional layer of protection, obviously. And then you see we have the complete database that contains on one side sensitive information and information that is intended to become public. And this last part, the public part, is also extracted, still in the restricted zone. We check that everything is okay, that it contains what it should contain. And this is this specific part, subset, which is pushed to our two public zones, and only this part. So like this, we have built something quite new. It's pretty original, but because we had to cope with very specific requirements due, again, to our partners, their needs, and the sensitivity of the data we hold. And now I think Daniel is going to guide you a little bit more and show you basically soon how our system looks like.
Thank you, Valérie. So one word on workflows. So essentially the public evidence is one of our inputs. We harvest it through harvesters and parse it so it fits into our SVIP model. We are in close contact with many of these resources, not only about licensing issues, but also about how we can improve API access or actually get rid of harvesters and make everything as seamless as possible, which may or may not mean also adapting the SVIP model to global standards. Once the data is, the public information is in SVIP, this can be accessed through our SVIP API, either through queries or through the SVIP user interface by typical Google-like search activities by researchers, which is basically the output. Now, we add this additional layer. What I said is kind of making us a bit different from existing platforms, is that we also gather actual variant data from our partner hospitals. This data gets then, as Valérie has explained, into a secure storage, and some aggregated data and the SVIP annotations, which is produced by the curators, which work kind of like over here. For some reason, this arrow has not switched up yet. But essentially, this information can go back to the clinicians. And as you see, not only do they have access to the public information, down here, they will have access to identify colleagues, which means they can see where a variant occurred. And if they make a query access through their hospital access through this secure client, they will get a DB snapshot report back. Basically, that means I am uploading a VCF and it will give us, it will return all the annotation we have for an uploaded VCF back to the clinician. This last part here, the secure client, the infrastructure is there, as Valérie just presented, but we are working now on this last mile of enabling a secure tunnel from the hospital into the SVIP sensor system, and also to have the reporting infra and the submission infra set up. So this is a navigation graph which I started using for the demos. So what I would like to demo you now, and this is maybe the biggest difference that we have had in the last 18 months of this project, that we now have a really nice system out there, which last October was published in its version 1. So what I'm going to show you is the title screen. I'm going to browse to the variant page and then look at some SVIP annotations. This is basically what researchers can access. Then in a second part, I will go to the curation dashboard, show you the curation interface for a single variant gene evidence item, and then a sneak peek at our brand new review dashboard. So I am switching now to the live system. So this is what you end up with when you enter SVIP.ch in your browser. As said, we have a nice Google-like interface that actually also can work with weird names. So your favorite gene name is usually supported. So let's start with Armadillo. When I just select one gene, I will get a list of variants. Note that in the beginning, I selected just SVIP variants. So this is why we only have 2 variants, because only for these 2 variants, we've already generated some curated annotations. So let's go here into S45F and look at the variant page. So here in the top part, you have a couple of general information about your variant. If a variant has a different transcript, it would be stored in a different variant page. So if your C coordinate is a different one, then it is handled as a different variant inside SVIP. I'm going to skip briefly the SVIP information and scroll down here to the public information. So here you can see the resources that we have ingested at the moment in SVIP. So for instance, if I look at what evidence items we find in CIVIC, I can see that it is focused on desmoid fibromatosis, so one disease. There are 4 evidence items. You can see 3 types of evidence: predictive, diagnostic, prognostic. We can see what the database entered as an output, as their interpretation of the variant. We have some overview graphic up here to summarize these events. We did not harmonize the level of evidence or significance from the different databases. In the public information, we simply give what these databases produce. So in case of CIVIC, we see the A to E evidence levels and how many we see of each of those. So this is one of our party tricks. We have a text mining functionality implemented in the SVIP-DB, which fetches the abstract from these references and highlights gene, variant, as well as disease, as you can see here in this example. If there are multiple variant references, we collapse them such to keep the overall overview of the variant page. You can, of course, always look at the specific different publications if you want to. Down here, we have some algorithmic impact predictions, polymorphisms, if available for said variant. We do connect to some other SPHN projects, amongst those the SVIP-O, which allows us to look at structural information of a protein. Of course, you need to select the right residue, otherwise you will not find the position of the variant. So this allows you to look at the molecule, zoom in and out. Let's go back to the central piece. So let's go for aggressive fibromatosis. If I click here, I actually see now what evidence items have been collected for this disease and said variant and gene. So this is a sortable table. So I can basically look here at what tier level we have. So professional guidelines, tier 1A. I can also sort according to the evidence type. So the typical evidence items, diagnostics, predictive, and prognostic are available, or we can sort according to drugs. Again, we can look at the publication. And if I click the publication, I will end up exactly on PubMed for this publication item. This is the public information which everybody can access on SVIP. And this is basically the pitch for the retrieval interface. I will now show you the dashboard for curators. So the dashboard for curators is not openly accessible. You need a login to get into the system. And as Valérie described, this is actually fended off from the outside world. So the dashboard has two main items: open annotation, so OnRequest Curations, and curation entries that have been submitted. They can have different statuses, so draft, saved, submitted, unreviewed, or reviewed. Reviewed, of course, being the highest level of existence an annotation can achieve. But let's briefly find our Armadillo variant. Oh, it's here. And let's look what happens if I go into the curation page. So for the curation page, we first have the first layer, which is the gene and the variant, maybe some additional information, if that is on a gene variant level possible, because afterwards a variant has different meanings depending on disease. If a variant allows, there can be a variant summary, and our curator's been super busy. So this is the most extensive variant summary I've ever seen one. If we go into history, we actually see who entered it when, and we can also see from which version it was changed into which version. So this is basically the whole system is fully auditable and traceable. Down here now, we basically see what type of drafts are existing for this variant disease. And we see this is basically always a different publication. So our curators, in this case, it's Anulka, she is drafting this version of the evidence item. But it's also possible that I want to actually add a totally new publication. Now, either I know a PubMed ID, I can enter it here and generate an entry, or I actually want to use our text mining tool. So the text mining tool basically grabs information from Medline, from clinicaltrials.gov and other resources, and looks for the occurrence of the gene, the variant, and the disease. And it also employs a clever expansion algorithm that allows to not only look for CTNNB1, but also all the other synonyms. It looks for positional information for different, so 3-letter amino acids, 1-letter amino acids, and so on and so forth. And based on the occurrence of variants and genes and diseases, it assigns a certain score to a publication. So let's assume that we want to add this item here. So I'll click here on the add button. And now I end up here in the curation interface. Again, before we had the dashboard, and now we are in the curation interface. So here we see the publication information. It's the abstract. We have extended this also to work with full texts. And down here, you basically see how our curators enter their information. So what we strive for is that we do not have free text evidence. Yes, I know there is a variant summary, And yes, I know they kind of like got us to add complementary information in free text, but we are a very collaborative team, you know. So up here, the central piece of the variant annotation is, of course, the variant. Then we can basically define what kind of what kind of disease we're looking at. You can even define the topo code, and you see everything with a red asterisk is required. We can also link up variants, so you can add additional variants here. And then you come here. So you can, for instance, select a predictive evidence item. The moment you select a predictive evidence item, the downstream classification changes. So it is asking you about a drug, which if you select a prognostic item, it will not do, because for prognostic items, you don't have drugs. So this is basically automatically changing the possible fields based on the top hierarchy selection, which makes the tool very lean and instinctive to use also for the curator. Once the information has been added, you can basically save a draft if you realize that there are other variants or other items, like for instance, there is a predictive and a prognostic evidence item in the publication. You can duplicate it because each item associated to a certain publication can only be one type. This is why we end up with fairly a lot of evidence items for each variant, disease, and gene triplet. I'm not going to save that now because otherwise I'll get into trouble. I'm going back to the dashboard. So here in the dashboard, what I omitted before was a brand new review interface. So this is basically what the curators prepare for our medical experts. They take a variant. So I have actually by heart learned where armadillo is supposed to be here. So we stay on this variant. So they prepare this and then they send this very small digest to our reviewers. Let me zoom out one spot. So this is what the expert will see. They will see a summary for this variant up here, you know. They will see the prognosis is unclear because we have good and poor outcome evidence items. So the annotation is unclear, while the clinical significance was tagged as included in professional guidelines. Now, this is what the annotators, the curators, suggest to be the SVIP annotation. The expert can either say, yeah, I agree, so they don't have to do anything, or they can see, no, it's clearly good outcome. The moment they change something here, their review status changes to disagree. And it is this moment where they actually need to provide a comment why they disagree with the annotation. They can add a publication and so on and so forth. And once they submit their review, it goes back directly to the said curator, and they can review and discuss with the experts on how they can change the annotation to suit the purpose. This is the same for all types of evidences. And essentially what you see here is your own status, and you would see here a gray symbol as soon as the other experts have made up their mind. They will not see whether they agreed or disagreed, but they will see that the review has been done. Okay, that concludes, and submit to review, of course. This concludes the demo, and I will hand over back to Valérie, who is going to speak about what's happening right now and what we're planning to do in the coming future.
Thank you, Daniel. Okay, so one of the things that was also another challenge was how do we structure ourselves, in the sense that at some point we are all handling data, sensitive data, and obviously we need to be compliant with the legal requirements, with the laws. And for that, you need a legal entity. So we are not the only ones. Obviously, this is a schema that is used for all the SPHN projects, I guess. But we are building currently, we are still in the finalization phase. We are building the consortium. So the consortium is the grouping of other partners, including ourselves as a PI, but here you see our clinical partners. And this is basically formalized through a document which is called the consortium agreement. And deriving from this consortium agreement that is acting mainly as an umbrella for the project, we have two other documents going on. One is the ethical approval. And this is a specificity of SVIP. We are not dealing with consented data. We are dealing with data that have retrospective data that have been generated for the care of the patients and that we want to collect and further annotate. And for that, we are currently close to submit ethical proposal for an exemption of through Article 34, the Human Research Act. So this will be covered by a specific ethical document. And the other documents that goes with the consortium agreement is what is called the DTUA, so Data Transfer and Use Agreement. It's an addendum, let's say, to the consortium agreement that is really defining very precisely how do we handle the data, what is done, by which means, etc. But it's very granular. And basically, why do we do that? That's clear. There's no order between the 3 points, but of course, we need to ensure compliance, whatever the law is. And we try to be compliant not only with the Swiss laws, but also with the European laws, because you will see that we target to expand our collaboration network. What is also very important, especially if we want to embark on a long-term project, we need to be very clear about the rights and the duties of all the partners, being us on one side, the technical side, I would say, and our clinicians. So this is very precisely defined in these documents. And in addition, we try to think in advance what could be the needs for new extensions or collaborations, including worldwide. And this is also described so that we are not stuck and we can collaborate in good conditions, well planned and well thought, so that we stick to our duties. Next slide, please, Daniel. One thing that I would like to mention, because this has raised, strange for us, I mean, we were not expecting that, but it has raised a lot of interest. So as part of the normal SVIP workflow, on one side, we will collect the VCF files that will be coming routinely, again, only retrospective data for the time being, and we will annotate them based probably on the number of occurrences. But pretty quickly, our clinical partners, they say that actually they would like something more. They would like to be able to request us to annotate for them a specific variant that they are interested in. This is what we call OnRequest Curation. So it means, apart from the VCF, when they will submit a file, they will be able to tell us, please annotate this one and do it quickly because I need it. I really need it to treat my patient. So this is a kind of parallel process. Next slide. That will be integrated into the normal workflow. So it will come back and will be available through the graphical interface. For the time being, this part of the tool is not ready for that. We are ready to do OnRequest Curation, but not to push that further. So because the clinicians wanted to have this pretty quickly, we have developed for the time being a parallel system, which is very basically through emails. But we are ready to launch this service in the coming days, probably. They can request us by email anything, assuming they are part of the SVIP project, obviously. And we will try to come back with a report, an annotation report, if possible, within 48 hours. That's our expectations. And we'll see how it goes. So we will start this pretty soon. And when we have presented this, it happened to us several times to present to international projects. And this specific, not only the curation interface as a whole, but this specific possibility to ask for curation has raised a lot of interest, and in particular from ClinGen, so the very well-known database, and the BRCA Exchange project. So we are currently actually working with them. And next slide, also with quite a number of other projects, mainly to understand where we can collaborate and how we can integrate better. Everything we did was planned to be integrated further with existing resources. But definitely we are working closely with the Global Alliance for Genomic and Health, the Beacon project, also we have a big part on that, and BRCA Exchange and ClinGen. Next slide. Basically, these are the main next steps that we are thinking of. We'll have 3 phases. First, the tool will soon become available for our clinicians with all the services going with it. And we definitely need to assess its clinical utility. Is it as useful as we hope? If not, what are the bottlenecks? And we will correct that so that it serves properly the clinical world, because that's our main objective, basically. So we will have some testing going on around clinical utility. We plan to further expand the global collaboration network that we already have, and not only the new ones that we want to the existing ones that we want to develop, but integrating with as many projects as possible, as long as it makes sense, obviously. And also we are thinking to expand this notion of on-request curation to other therapeutic areas, because again, we have seen that it's raising such a huge interest that we think that eventually our annotators are absolutely able to annotate in additional diseases. So we are thinking to expand this functionality. On the next slide, I will simply show you not only the partners we have, so the hospitals that are really partners in the SVIP consortium, but also we collaborate with other SPHN projects, and namely SOCIP and SVIP-O. And we have, since the very beginning, I must say, we had the strong support from the Swiss Society of Molecular Pathology and the Swiss Society of Pathology, which was really great, because without a professional society behind us, it would have been more complicated. So we are pretty grateful to all those people for their strong support, and also clearly our partners, they are very committed. We ask them quite a lot, actually quite a lot of input, and this is how we build the system, and they have been always very proactive and very positive about the project and their support. So we are also very grateful to them. And on the next slide, so these are the people practically doing the job. So basically the team of Daniel at ETH Zurich, the team of Patrick Ruch, who was with the text mining group at the HES, and my group in Bioinformatics, and people from the Swiss Prot group, who are the one taking care of the annotation. Next slide.
So maybe I chip in here. So we do distinguish between 2 things in SWIP. So when we speak about the SWIP service, we do mean SWIP.ch and its API that is instantiated and built for our hospital partners. But the whole infrastructure, the code, the source code of the whole infrastructure is publicly available on GitHub under GPL free license. So when we speak about global collaboration and extending to other therapeutic areas, this is not only focused on the curational efforts, which flagged most of the interest, but also that people would like to leverage on the existing curation review interface that we have built. So there we see great opportunity, why we have it as one of our 3 main legs in the next steps. So we are happy for you to basically browse to SWIP.ch, look at our infrastructure, feed back to us on general stuff, If you happen to dive into the code, please submit issues on GitHub. And ultimately, if you're a partner, there is curate.svip.ch, and this is going to go live soon. And thank you for your attention.